Good morning, students. Students, as we discussed about cancer in our previous video, as I mentioned that cancer is a kind of abnormal growth of any kind of tissue or any kind of cells. And I told you that cancer may be of two different types. Either it may be benign tumor or maybe malignant tumor. Now today I want to discuss different causes of cancer. And I will also tell you that how can we diagnose cancer and how can we prevent ourselves or how can we treat cancer. So today I am going to start from the topic what are the different causes of cancer. Students, I want to tell you that there are various agents which can cause cancer, and these agents which are causing cancer are called as carcinogens. Means the cancer causing agents are called as carcinogens, एंड दे मे बी ऑफ डिफरेंट टाइप्स. तो ये कार्सिनोजन जो है डिफरेंट डिफरेंट टाइप्स के हो सकते हैं. नाउ इनिशियली आई एम टॉकिंग अबाउट फिजिकल एजेंट्स. नाउ व्हाट आर फिजिकल एजेंट्स? फिजिकल एजेंट्स इंक्लूड ionizing radiations like X-ray, gamma ray, or as well as non-ionizing radiations also can cause cancer like ultraviolet rays. So these radiations, either these are ionizing radiation or non-ionizing radiation, either these are X-ray, gamma ray or ultraviolet rays, can cause DNA damage and leading to the neoplastic formation. Means these radiation can damage our DNA, and once our DNA is going to be damaged, so it will lose, or due to it the cell will lose the capability of contact inhibition and they will become neoplastic. What is the meaning of neoplastic? When a cell is losing the capability of the contact inhibition. What is contact inhibition? A cell regulates the growth of neighboring cells, or it doesn't allow neighboring cells to grow, or मतलब अनकंट्रोल्ड ग्रोथ को एक कंट्रोल करने का काम करती है. इज इट क्लियर? नाउ द अनदर थिंग इज this is why the indiscriminate practices using x-ray should be avoided. So that's why you are always suggested that please do not go for x-ray and do not sit at that place where the x-ray is going to take place, because these x-ray can damage your DNA, and due to the damaged DNA your cells can lose the capability of the contact inhibition, or they can convert into the neoplastic cell. If they will convert into neoplastic cell, so it can be cause of cancer. Now the second kind of carcinogens or agents are called as chemical agents. Now you all most of the cases you are oftenly observing that in the packet of the cigarettes as well as the tobacco, it is mentioned there tobacco kills. It means tobacco as well as the smoke of the tobacco is the major cause of lung cancer or throat cancer or mouth cancer. ठीक है, because these chemicals can convert your normal cell into a neoplastic cell. Similarly, some chemicals like benzene fume can also cause different types of cancers. Now the third one agent which is also responsible for cancer is called biological agent, and basically there are some viruses which can cause cancer. Actually those viruses which are causing cancer are called as oncogenic viruses. Means a virus which causes cancer is referred as oncogenic virus. They have genes called viral oncogenes. Certain other genes called cellular oncogenes or proto-oncogenes have been identified in a normal cell, which when activated can lead to the tumor formation. So I want to tell you that there are some viruses which are having some special type of genes, and as we know that whenever virus attacks on a normal cell, it captures all the machinery of a host cell. Means it introduces its DNA into the host cell, and its DNA becomes the part of the host cell DNA. So it means it has DNA which are having some genes. These genes are called oncogenes, and these oncogenes will become the part of the normal human DNA. So due to the presence of these oncogenes, the DNA will mutate. The DNA can mutate, and it will, due to it the cell can become neoplastic. And I want to also tell you that in sometimes in our normal cells the DNA contains some genes. These genes are referred as oncogenes or cellular oncogenes or proto-oncogenes, because these genes are already present in the cell or cell's DNA, but these are not in activated form. So due to certain regions, when they are becoming active, so they can cause different types of tumor too. So in this way we discuss three types of causes: physical agents, chemical agents, and biological agents. Now I want to discuss how can we detect cancer or how can we diagnose cancer. So cancer should be detected in the early stages. ठीक है, if it is detected in the early stages, then the treatment is successful. ठीक है, and it can be detected by the various methods. Now what are these? The first one is blood and bone marrow test. ठीक है, we can count the number of cells in our blood or bone marrow, like in leukemia, and we can diagnose cancer. ठीक है, तो in blood and bone marrow test we are going to count the increased number of cell counts like in leukemia. Now second test is called biopsy. In biopsy, one piece of suspected tissue is separated out from the body. ठीक है, and the sections will be cut from that particular piece, and then we will strain them, and then we will examine them under microscope, and then we will find out that are these cells are normal or abnormal. Means we are taking out the suspected piece from the body, and then we will cut it into sections. ठीक है, then we will stain it, and then we will observe it under microscope, then we will make a comparison from a normal cell. ठीक है, तो we will definitely able to identify are these cancerous or non-cancerous. The third technique is called as radiography. Okay, in radiography, X-ray is done to detect cancer of internal organs. With the help of the X-ray, ठीक है, we can detect the cancer if it is present in the internal organs. Now the fourth one is called CT. CT means computed tomography. So in computed tomography we have to generate 3D images of the internal tissues. ठीक है, विद द हेल्प ऑफ द एक्सरेज. तो कंप्यूटेड टोमोग्राफी में जो इंटरनल ऑर्गन्स हैं उनकी 3D इमेज को कैप्चर किया जाता है और उसकी एनालिसिस की जाती है. फिफ्थ वन टेक्निक इज कॉल्ड एज मैग्नेटिक रिजोनेंस इमेजिंग. तो इट इज़ कॉल्ड एज MRI, मैग्नेटिक resonance imaging. It involves the use of non-ionizing radiations and a strong magnetic field to detect pathological or physiological change in the living tissue. So basically in MRI we are going to use non-ionizing radiation as well as high magnetic field or strong magnetic field. With the help of it we are going to identify or to detect is there any pathological or physiological change in a normal cell. If there is a change, so we can consider it as a cancerous cell or cancerous tissue. Now the last technique is called monoclonal antibodies test. Monoclonal antibodies against cancer specific antigens are also used for the cancer detection. Already prepared antigens or antibodies are introduced in the body to detect cancer. ठीक है, so in monoclonal antibodies test we are introducing some antibodies which are cancer antigen specific, and with the help of it we can identify is the cell or the tissue is cancerous or non-cancerous. Now these are the 6 different techniques through which we can diagnose either the cell is cancerous or non-cancerous. Now finally I am talking about treatment of cancer. Now how can we treat cancers? So most of the cancers are treated with a combination of procedures like surgery, radiotherapy, chemotherapy, and immunotherapy. So means either we can go for a single procedure, or we in most of the cases we are applying the combination of the procedures like surgery, radiotherapy, chemotherapy, and immunotherapy. तो सबसे पहले बात करते हैं सर्जरी की. In surgery, the tumors are removed by surgery to check further spread of cancer cells. So initially the tissue or the tumor which is suspected of cancer, we are going to remove it out from the body, and then finally after that we are checking is the tumor is spreading or is it okay. ठीक है, the second technique is called radiotherapy. In radiotherapy, tumor cells are irradiated by the lethal doses of the radiation while protecting the surrounding normal cells. So in radiotherapy, because we are going to burn the or we are going to remove the particular cancerous cell with the help of the radiotherapy, and one thing is noticed in this process, that other cells which are surrounding to the cancerous cells should not be harmed. ठीक है. Now the third therapy is called as chemotherapy. In chemotherapy, several chemotherapeutic drugs are used to kill cancer cells. So various chemicals or chemical drugs are used in such therapy to kill cancerous cells, but the people has too many side effect of chemotherapy. They can have hair loss as well as anemia too. And the last technique or last procedure which is applied to treat cancer is immunotherapy. In immunotherapy, biological response modifiers like alpha interferons are used to active the immune system and help the destroying tumor. So generally we know that when a virus infects any cell, so this cell starts to release interferons. So similar kind of interferons are introduced in the body, like alpha interferons are used, and with the help of these alpha interferons, the other cells of the body are in alert that they will not be in effect, they will not be come in effect of that particular virus which is going to damage our cells, and we can protect our cells from the cancer. Okay, so in this way there are four different techniques: surgery, radiotherapy, chemotherapy and immunotherapy. So I hope so you understood the topic very well. Once again I will meet you in the next video along with a new topic. Till then, thank you, bye bye, and have a nice day.